All right, let's continue with this. Okay, before everybody is coming, how many of you there? Nathan? Can you hear me, Nathan? If you can, give me a thumbs up. All right. Nathan, what are homologous chromosomes, Nathan? And still dreaming. Who would like to help him? Gwen?
Homologous chromosomes
Yeah.
are chromosomes made of two of the same sister chromatids, right?
Same sister chromatids?
Yeah.
Hmm?
Mm-hmm.
Anyone else would like to help them out?
Is it it has 46 pairs of chromosomes?
You can say that a set of chromosomes which has got one maternal and one paternal chromosome.
Okay.
So there are a set of chromosomes which you can see on your screen, which has got one maternal and one paternal chromosome. These are homologous chromosomes. When it will form a sister chromatid with it, which will be called a tetrad. Again, a set of one parent, this maternal chromosome and a paternal chromosome are homologous chromosomes. Okay, so this homologous pair, this one and this one, these are one pair, they will synthesize or they will replicate. Once they replicate it, they form their sister chromatid, which is connected at the centromere. Okay. So these are the sister chromatids. They are still called homologous, but they are tetrads because there are 4 chromosomes there. So they are also called tetrads. Okay. But the sister chromatids will be connected at the centromere. Clear? Difference between homologous chromosome, sister chromatids? Now, in prophase 1, DNA is going to supercoil, chromosomes are condensing, nuclear membrane will dissolve simultaneously, centrole will start moving at the different phases, different ends of it. Then there will be a production of bivalent. What is bivalence? That is during the synapses, okay? So the homologous chromosome associate with each other to form bivalence. Bivalence is only when non-sister chromatids, these are sister chromatids, they're identical to each other, okay? This is the mother and this is the father. So the chromatid of the mother and the inner chromatid of the mother and the inner chromatid of the father. Why am I using the inner word? So that you get clear idea. They will cross over, the alleles or the fragments of the chromosomes will cross over. When this crossing over is going on, they are called bivalents. Okay, so this will be called as a bivalent because there's a crossing over taking place. Now this crossing over is shown only at one position over here, but it is not necessary that will take place only at one position. There could be many crossing overs will be formed on this chromosome. Okay, So the crossing over between non-sister chromatids takes place, and this results into the recombination of alleles. So recombination of alleles means the alleles which are found, suppose that this one has got a dominant allele and this one has got a recessive allele. So it will get exchanged between them, okay, which will cause the genetic variation. That's why each child is different from one another, except when we are talking about twins. But if you look at your siblings also, or you look at your brother and sister, each one of them will look different from one another because of the genetic variation in the gametes. Clear? In fact, you might be thinking that this genetic variation is taking place and you're talking about this variation only in human beings, but it happens in all the eukaryotic cells, wherever there's a sexual reproduction taking place. So even if a tiger cub looks similar, but they are different from one another. Okay? Even if we are looking at the plants where the sexual reproduction has taken place, each one of is different. That's why Mendel was able to work with the peas, because whenever there was cross-pollination taking place, he could have a different variant which could be seen. So leaf can be rough, but had purple flowers. But the parent would be different, having white flowers. So variation could be seen between them because of this crossing over. Are you guys with me? Thumbs up if you're with me. Okay. Now these are the bivalents which are formed, but here they have not shown the crossing over, but it will be only when the crossing over is taking place, they will be forming a bivalent. Please remember that. So these are the bivalents which are formed. So the point of crossing over between the non-sister chromatid is called chiasma. Okay, or there would be many chiasmas taking place. It means here the crossing over is shown only at one place, but there would be many of them which are taking place, so there would be many chiasmata. So the point at which the crossing over between the non-sister chromatid is taking place is called chiasma. Please note it down. And this will lead to the recombination of different alleles. Means different variation. So suppose this one had for color blindness. Now color blindness is recessive if you remember. So if suppose this is for the color blindness and this one has the dominant one which does not have the color blindness, when it is getting crossed over, that's why there is a possibility that one child will have color blindness, even if it's a recessive. So the recessive has been exchanged over here. Now chiasma, if you look at this, here there is one chiasmata which has been, chiasmata which has been taking place. But look at this one, many different patterns are formed because there are many chiasmata.
Ma'am?
Yeah.
Can you go back? I didn't finish taking notes.
Okay. Anyone else taking notes? I will be sharing this in Google Classroom.
Done.
Done? Okay. So how many chiasmata can you see here? 1, 2, 3, 4, 5. 5 of them are taking place over here, so at different sections of it. So chiasma occurs between the sister chromatids, therefore should be appear at the central space. The formation of chiasma is called common and is thought to be essential for meiosis. It has to take place in the meiosis. If it does not take place in the meiosis, and remember one thing, mutation can also occur at this point also, when the proper division is not taking place. So chiasma occur between the sister chromatids and they should be appear in the same way. The first two, and here you have 5 chiasmatas. Now, it increases the genetic variation through combination of linked alleles. Now remember one thing, one trait is, some of the traits are having many alleles which are responsible for one single trait. Okay, many genes are there which are responsible for one single trait. So suppose you are talking about the color of the hair. So there would not be only one particular allele which will be responsible for it, but there would be many of them which will, many genes which will be responsible for the character of that particular trait. Okay, so they are called linked genes. So these are genes which are linked together for one single trait. So here, when the synapse is taking place between the homologous chromosome, the chiasmata is performed between the neighboring non-sister chromatids are cut at the same point. This is called a holiday junction. This one is called a holiday junction, forms as a DNA of the cut section attached to the open end of the opposite sister, non-sister chromatid. So this junction which is formed between them, between the bivalents, is called a holiday junction. And this will cause a recombination. Now, if you look at this, the capital E is showing you a dominant character and the small e is showing you recessive. Now, this one has got, suppose this father has got the dominant characters and the mother has got the recessive one. Because of the exchange or because of the crossing over, what happened? There was this particular chromosome has got the allele. This, because of the swapping of the allele, it has got the one dominant allele here, and this one has got the recessive. So if suppose most of the characteristics which are responsible here for the father, from the father's chromosome, which a child has got this one, this person might look like a father or might behave, some of the characteristics will be like father, but can have this particular allele which is recessive. So increases genetic variation through recombination of the linked genes, which we already spoke about. So this one has got one dominant, one recessive, and because of the recombination, because of the variation, they were changed. That's why each one of the chromatid is different from another one. So crossing over leads to more variation in gametes, and this is a standard. Okay. This is again the same thing when you are drawing. If you have been asked to draw, please use color of shading to indicate each of the sister chromatid. Remember, you will not be allowed to use different color pencils. But what you can do is use pencil and pen and to show it, or you can just shade it to show chiasma and plus recombinant chromatids. Now in metaphase one, what happens? The bivalents which are formed, remember once the exchange of the genetic material is taking place, they are called the bivalents. The bivalents are formed here at the equator. Now this is in one cell. So it might happen that this one can go on this side or this one can come on this side. Or it can happen a change over here. So there would be a random orientation which will take place. And this will divide them, this will separate them into a different daughter cells, the daughter nuclei. But this will be a random positioning of the equivalent which is taking place on the equator. Okay, so random orientation occurs and each bivalent aligns independently and hence the daughter nuclei gets a different mix of chromosomes. So there would be a high variation. Now there are 2 chromosomes and there are 23 variants, if you remember. The 2 power 23 in human beings, you will have different combination. So these 2 chromosomes are aligned like this, but here it might happen this has swapped places. Okay, there would be another chromosome where these 2 are swapping places and these 2 are in the same position. So there could be different variations which are taking place over here because of random orientation. Are you guys with me? How many chromosomes are there? 23 pairs are there. Okay, they can just jumble up anywhere. If you're looking at a sequence, it does not form like your alphabets. When you're looking at alphabets, happens in sequence: A, B, C, D, E, F, G, H, I, J, K, L, M. Okay, all the way till Z. But there is a possibility when you're talking about random orientation that after A, it can be a Z. Okay, so there are different variation which can be formed over here. Is it too much? Alice? Getting it, darling?
Yes, ma'am.
Eliana, Keita, Dika, Basia? Okay. So allele have a 50% chance of moving to a particular pole and the direction which, now if you look at this one, there's something which what I was talking in your previous slide, that there are different orientations which are there, and because of that there is a random orientation, and because of that there would be many variations which are taking place. So therefore different alleles combination should always be equally possible. If the gene loci are on different chromosomes, this does not hold for the linked genes. Now the linked genes, which it has to have the genes which are responsible for a particular trait, it has to exchange at the same particular loci. If it doesn't, then it will cause, it cannot happen on different chromosome. It should be between the non-sister chromatids only, so that it does not have variation in the linked genes. Now, in anaphase 1, the homologous pair will be separated, but they will remain attached on the centromeres. But in metaphase 2, these are getting, we are jumping from metaphase 1 to metaphase 2. In metaphase 2, the sister chromatids are getting aligned. Okay, when the sister chromatids are getting aligned, the spindle fibers will connect to the kinetochores or the centromeres. And at that time, the sister chromatids will be separated from each other. So, pair of sister chromatids align at the equator and spindle fibers to form and attach to the centromere. Random orientation again contributes to the variation in the gamete, though not as such at the extent of metaphase 1. There's not much random orientation because the possibility is less over here. Over there, the possibility was high because there were many chromosomes which were present. But here the possibility is little less because the number of chromosomes have reduced, because there are only sister chromatids which are there present. So this is because there's only difference between chromatids when crossing over is taking place. In anaphase 2, what will happen? The spindle fibers will start contracting. When the spindle fibers start to contract, their sister chromatids will separate at the centromere. And if you look at this one, this will form one cell, another cell, third and fourth. Each one of them is different from one another. So due to crossing over, each of the 4 nuclei is likely to be genetically different. Cytokinesis begins, splitting of the cell, end of the meiosis is 4 haploid genes. Okay. Are you guys okay with this? All right. Let's check your language. What are these? 4 separate chromosomes. Are there 4 separate chromosomes?
Bivalent.
Bivalent. They are bivalent. Very good. What is this?
One pair of sister chromatids.
Very good, Anga. One pair of sister chromatid. Okay, I want somebody else to speak now. What are this? Okay, why aren't, okay, tell me what is this one?
2 separate chromosomes.
2 separate chromosomes. Why aren't they homologous chromosomes? Why aren't we calling them homologous chromosomes?
Because they don't have the same allele.
They don't have the same allele, plus they are different sizes altogether. So they are 2 separate chromosomes. What about these?
Two bivalents.
They are 2 bivalents. Very good. Okay, this is just a summary which I will be sharing it with you. Are you guys clear with this? Now for HL, what I would suggest is go through page 415, 416, 417 till page 420 for HL. Okay, SL, what you can do is you can continue with your IA, remaining 15 minutes. All right, BSc students, you can do any of your pending work, whatever is left out, like the last paper is left out, please complete that, and HL will read. We will discuss doubts tomorrow. I want the SL to join again tomorrow for another class. Okay?
Okay.
All right. Giving you last 15 minutes to read. Come on. And if you have
Ma'am,
doubts, yeah.
Ma'am, can you check the WhatsApp?
All right. All right, I'm ending this Zoom session right now. If you have doubts, send me on WhatsApp and we will discuss it tomorrow. Pasya?
Yes, ma'am.
I think I've written some comments for you in your IA.
Okay, I'll check.
Please go through it. Okay, bye guys.
Thank you, ma'am.
Bye.
Thank you, ma'am.
Please read, because tomorrow when you're coming back to the class, I want you to ask questions. Or I will ask questions.